Okay, so our next speaker is Avais Farid from Hong Kong University of Science and Technology, and we'll talk on a new antimicrobial coating system for long-term caring facilities for elderly in Hong Kong.
Thank you very much. And I'm Dr. Farid from Hong Kong. I'm professionally medical doctor, but interdisciplinary program office of an engineering university. Hong Kong University of Science and Technology gave me opportunity to work something in chemical engineering and apply that in medical field. So the topic that I'm going to present today is a system that is antimicrobial against the bacteria, especially present in the hospital environment. So this system is gonna kill those pathogens that are harmful for the human health. So I would start with some background. The topic is about elderly. So how serious is this thing? Globally, the statistics is telling that the population of elderly more than 60 years of age is going to be double in 2050 from 9-10 million to 2.1 billion. And as this research we have done in Hong Kong, so I want to share that population of old people more than 60 years of age will be 2.6 million, like around 36% people will be older in Hong Kong in 2066. So that was one issue, like this population is more vulnerable to acquired infections that may cause serious illnesses and deaths. So OECD says that around 700,000 deaths occur yearly because of these infections, and drug resistance is also an issue. So keeping in all that thing, it's not only about health, but also adding the cost. So every year, 10 million US dollar is adding up, and the cost will be around 100 trillion US dollar with the passage of time. So coming back to the situation of Hong Kong, the situation is not very clear, not very satisfactory for the infection control. The media reporting that these superbugs are infecting every 18 minutes some residents, some people in the hospital settings, and it's not under well control. So the number of these infections are increasing and the registered patients are increasing. So hospital authority just showed the statistics for 5 years, so they couldn't eliminate these infections, although they are reduced with the time, but not much, not significantly. But we did a pilot study when I was starting this project, that we need to first figure out the environmental burden of disease, how much bacteria are in the environment. So this study gave us an idea which type of bacteria are in the hospital settings. So here we find out like most of them are gram-positive, in which few of them are negative, but we were focusing on the most notorious one, MRSA, or MRSA you call it. So that was there around, we find 10 raised to power 5 CFU per meter square. So that was quite interesting for us to work on this area. Keeping in mind all these things, we plan in a way that we need to prepare a system that will be very effective antimicrobial system. We need to have some safety things for optimization of and concentration of the coating. Then we will do some pilot study to calculate the sample size and also see the settings in the clinical environment. So our final plan was to do some double-blind clinical trials so that we can have the final conclusion about the technology and the results. But all of these things keeping in mind, we wanted to make something that should be inexpensive, easy deployed, and consistent, like giving a good result with the time. But also wanted to add the ingredients that must have some safety approval, already approved from US EPA and US FDA. So this coating, the plan was should have some contact killing, like immediately bacteria touching to the coating surface should die at the time. And it shouldn't have any anti-adhesion properties. It may prevent some surface colonization and also not prevent some biofilms. And the temperature is changing, in the weather is changing, so it should have good tolerance for high temperature and low temperature. So all that we have done, we made a chemical coating, and that one I got patent in 2016 for this one, for 1 year provincial patent, then it was permanent, and now it's in 4 countries. So briefly, I would like to show you how this antimicrobial coating, that's basically polymers of in a micellar form, 3 cores. So 2 cores have this chemical like polyethylamine and polyhexanide. So this is a picture that you can see some micellar form at 100 degrees centigrade. I just took this picture for size was around 10 nanometer. So just for your demonstration, it is like anti-adhesion properties and antimicrobial property. So this technology we developed in 2016. Then starting with that, I started some lab testing, getting some clinical isolates, really pathogenic bacteria, mostly drug-resistant strains, to check in the control environment. So the plan was to deploy this technology on the porous surfaces, especially the hospital curtains or bed sheets or gowns. So this area still had some space to work on. So in the lab, in the control environment, the test we have done, so I'm just giving you summary, that we give challenging time of 1 minute. So we wanted to see the bacteria are going to die with this polymeric technology in 1 minute. So we find around 99% bacterial cells were dying. So here are some bacterial strains that are drug sensitive, like Staphylococcus aureus, Enterococci, and Pseudomonas E. coli. But I also checked some drug resistant strains like MRSA, VRE, drug resistance, Acinetobacter, and Pseudomonas. So and also the sustainability, keeping in mind. So these results are for 30 days. For example, if I coat a piece with the coating, so after 30 days continuously exposure of bacteria to the surface, so what will give the results? So we find around 90%, minimum 90% reduction in this control environment than in the lab. So the summary of this one was for the drugs resistant and drug sensitive one individually. So this is the reduction at day 30 with the 1 minute contact time. So we find around more than 95% reduction in for drug sensitive and drug resistant strains when the bacteria were exposed to this chemical coating. So keeping in mind all those things, the objective of this clinical trials we planned in further was that we need to assess this technology, its performance on the porous surfaces. So we selected the bedding material, but this time we did it on in the elderly home. It's a local elderly home from the government. So we put this coating on the bed sheets. So the results I will show in the future. So the target was the bedding material, and we needed to check the total bacteria without discrimination of which bacteria is this, using some agar plates. And the specific drug resistant strain was MRSA. And also need to assess something like what other type of drug-resistant or drug-sensitive bacteria are found in that environment where the elderly people are living for their life. So time frame was like, it was, 4 weeks we have taken the samples starting from January till March. And we had two ends of this clinical trials, one on control and one on treatment. So from different beds, from different floors, we collected the samples in different observational period. So in 4 observational period, we divided this study and collected the samples from the bed sheets. So for more understanding, like the idea of this coating deployment was to put this coating at the time when we put the fabric softener, when already linens are clean, so we put it at that phase. But to make it easy, we just use the dipping method. When it's washed, just dip it inside, give 10 minutes time to absorb this chemical coating, then just squeeze extra liquid and put it in the dryer, and those are ready. So that was the coating method of our chemical coating on the bed sheets and curtains. The methodology is the study design was double-blind crossover interventional study, because it has 2 arms. So on that one, we, as I mentioned, that we wanted to check the total bacteria. For that, we use tryptophan soya agar. And for MRSA, we had a selective media, it's called chrome agar MRSA. For sampling, we use a standard poly wipe sponge, and we took the sample randomly from different sites. And the samples were later on put on the neutralizing solution to neutralize the reaction. Then we did the inoculation, like putting samples on the plates and then counting them after 24 to 48 hours to count the colonies of bacteria. So for brief way, I will say we divided one bed sheet in 3 areas, upper, middle, and lower area. So this is the sampling area we have done. So in this area, we can take samples. So location was like, call it heaven up of Whooping Care Center. So ideally, 258 samples were required. We calculated statistically, but we took a little bit more samples from 94 bed sheets. But all of them were from randomly and blind. Only the principal investigator, like I was the principal investigator, so I know which sheet is coated, which sheet is not coated. So the team was randomly taking the samples. We have coding on it, and we take the samples for 4 consecutive weeks, and 3 samples from each site. So we do the cultures of the samples and do the enumeration. So this is prepared because it's the environmental sample. We don't need biosafety cabinet or anything. So we put those on the desks. So when we have done that one, so autoclave in normal way that we usually proceed. So we'd like to share the results actually for total bacteria count without any discrimination of resistant strain or not. So we found around 80% reduction from control to treatment. And this was the results of total bacteria. And on MRSA, we found around 86, 87% reduction. So it was around more than 1 log. That was interesting. So the infection control team and hospital authority was quite happy with the results for this reduction they have found on that. But it was interesting, like you can see in the control there was around more than 5, and here it was more than 4. That was pretty alarming that time. So the consistency of antimicrobial efficacy we have done for 4 weeks. So once we put it in the coating material, so we did not change it for 4 weeks. So every week we try to take the samples and check the reduction, how much reduction is happening on the treatment end. So it has given us around more than 80% reduction on this. On this slides you can see the. And statistically we wanted to see the results are significant, so we use some t-test for variance of 2 means, and for upper, middle, and lower area, because there were more samples, so we have done ANOVA. The results were good because p-value was less than 0.000, so pretty significant. So the summary of this clinical trials, the results we have done in elderly home. So this technology has given us very good results because they have showed very good antimicrobial efficacy against common bacteria and pathogenic bacteria and some MDROs. and it has proved its durability. In the laboratory testing, we have tested for 3 months, but in the clinical settings, we have done for 1 month. But it has proven very durable and long-term surface antimicrobial thing. And it has showed you, it has maintained 70%, more than 70% reduction, and it decreases the viability of microorganisms on the specific surface where that was coated. So as a consequence, I will say it is beneficial and putting a very good impact in reduction of unwanted microbes. So this kind of coating on specific surface, we can coat with that and can get very good antimicrobial results for this kind of technology. Thank you so much. So you're welcome to ask the question.
Yeah, thank you. So not much time to have some questions. Maybe we can skip that part,
yeah.
And or maybe one. If you have one question, what burning question?
Yeah.
What is the safety of these materials?
Yes,
because
okay.
supposedly they'll be in contact with skin, and
Yes, yes.
and not all bacteria are bad. The
Yeah.
natural flora is good for people. So I would suppose that these materials would kill also the natural flora of people. No?
Right. Okay, for the safety, there's 2 types of testing we have done. MTT assay for 3 cell lines, lungs, kidney, and skin. So initially, the concentration, like it took 2 years to optimize the safety concentration we have reached. So this coating now we have using the concentration is safe, that the cells in the inhibition was not more than 50%. So the 3 cell lines give us positive results that this is not killing the skin, kidney, and lung cells. Secondly, we have done volunteer skin testing. That is usually the beauty products they do. They put the samples on your skin for 72 hours, and we assess any sign of inflammation. So that one we have also done. So with a shortage of time, I couldn't put that as safety one. And this is the second question is like, yes, you're right. But we try to focus more in the hospital environment and the elderly. They are having this because for that reason we have studied. We have done a pilot study to figure out which type of bacteria are there. So we find more MRSA and mostly gram-positive bacteria. But right now, we don't know what beneficial bacteria are dying with that. But the target was to kill these pathogenic strains, and till now we are successful. But hopefully with the passage of time, we can further add something that cannot kill some and kill the targeted bacteria.
So let's move to our next speaker, and thank you.
Okay.
Thank you once.